Hello and welcome to Genetics and Inheritance Part 2 revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. What is genetic probability? Probability is used in genetics to predict how likely it is that offspring will inherit certain traits from their parents. By looking at the alleles each parent has, we can calculate the chance of different genetic combinations using tools such as Punnett squares. So for example, a probability of 1 in 4 or 25% means that, on average, 1 out of 4 offspring is expected to show that trait. Single gene crosses, where only one gene is involved, are relatively simple to predict, but there are only a few possible outcomes. However, most characteristics in real organisms, such as height or eye color, are controlled by multiple genes working together. This makes their inheritance much more complex and less predictable than single gene traits. We can use a Punnett square to investigate inheriting height in plants. So a big T is the allele for tall and a little t is the allele for short. If both parents are heterozygous, so we've got a big T, little t. When we put down the genotypes of the parents, we can then fill in the Punnett square. So the big T and big T makes big T, big T and little t, and then finally little t and little t. And overall the offspring will end up being 75% tall, so that's big T or big T, little t, and 25% dwarf, which is little t, little t. In a different example, if one parent is homozygous dominant, big T, and the other is heterozygous, big T, little t, that means that our genotypes will be big T and big T, little t. Now when we come to fill in the Punnett square, again, big T and big T will make big T, and then the other combination will be big T and little t, which makes big T, little t. So that means that 100% of the offspring in this case will be tall, some will be homozygous dominant, others will be heterozygous. If one parent is heterozygous, big T, and the other parent is homozygous recessive, little t, then our genotypes will be big T and little t. We can then fill in our Punnett square. So we've got big T and little t makes big T, little t. And then we've got little t and little t makes little t. And that means that our offspring will be 50% tall and 50% dwarf. In another example, if one parent is homozygous dominant, big T, and the other is homozygous recessive, little t, that means our genotypes will be big T and little t. And we can then fill in our grid, and it means that all of the offspring this time will be big T, little t. So that means you've got 100% tall, big T, little t. Thank you for watching. See our revision notes, the link is in the description.